It's my pleasure to introduce Wim Vanduffel. I have worked with Wim for several years. We are very good friends and colleagues. Wim is the head of the research group Neurophysiology and of the laboratory Neuro and Psychophysiology at the Faculty of Medicine at the KU Leuven, Belgium. In addition, he still holds a PhD at the School and is affiliated as researcher at the Massachusetts General Hospital. Wim studies visual perception and cognition in human and non-human primates. He has pioneered awake behaving monkey fMRI techniques and used it to investigate functional correspondences between the human and macaque brain. By combining fMRI with state-of-the-art causal techniques, his lab aims to study interactions between retinal and non-retinal internally generated signals and the mechanisms driving adult cortical plasticity underlying perceptual learning. Lately, Wim's group engaged in developing novel, very high resolution imaging methods to study the functional organization of the brain at mesoscale level. Okay, Wim, you can start your talk.
All right. Thank you very much, Reza, for the very nice introduction. Yeah, it's a pity that I cannot be there in Iran. I'd love to come, but unfortunately, our administration didn't allow me to travel there given the circumstances in the Middle East at this moment. But for sure, at some point in the future, I would love to visit you guys there. And our yeah, I know Reds actually for a lot from the time that I was very frequently in Boston, and I collaborated at that time with the PI of Reza, Roger Tutel, and I think we did very nice work in these days, which basically inspired me to do whatever I'm doing right now. I would like to start by thanking the two people in my lab who did most of the work, if not to say all of the work. Chi Zhu and Chaolian Li. They're also a couple. Chi Zhu has now an independent position at Neurospin in Paris, and Chaolian unfortunately left science despite the fact that they think she was a brilliant researcher. So our current thinking about the architecture, the functional architecture of the monkey cortex is one where we consider the fundamental building blocks being areas. And areas, cortical areas, as we all know, are defined based upon function, cyto and myeloarchitectonic features, connections, and whenever relevant, of course, topography. For example, with the topic organization, visual cortex, tonotopic organization, and auditory cortex, and so on and so on. And based upon these criteria, we all know this very, very famous, probably the most used image in systems neuroscience over the past decades from David Van Essen and Velleman, which shows an overview of the cortical areas. And of course, since 91, it has been modified a little bit, but not to a great extent, where you see all in color visual areas and other cortical areas, which are not colored here on this flat map of the macaque brain. The influential work of David, of course, not restricted itself to the layout, sort of like the functional layout of the cortex. He also identified connections, not only by the work he did himself, but by collecting data from many different laboratories, where he sort of like put up this connectome, where you can see this very complex diagram, again, which has been modified considerably since the 91, where you see the connections between the different areas, connections that provide information to higher order areas, feed-forward connections, but equally so connections that provide feedback information from higher order areas to lower order areas. Do you see my, I'm gonna use my pointer basically, basically from higher order areas to lower order areas. The big problem with all, despite the fact that it's of course very, very influential and very, very useful, the big problem with this diagram, and actually most of the connectomes that we currently use, even the adapted ones, is that individual areas are considered as singular points. It is as if, for example, V3 is one single homogeneous area that then connects to different other cortical areas. We can summarize this in a diagram like this, where areas are visualized as boxes here, and with the feed forward, feedback, and also the lateral connections between areas at the same hierarchical level, sort of like the same view as what you have just basically showing the hierarchically structured network of interconnected areas. This is how many models of the brain basically currently, or many models of the brain are using this schematic. I think this is an utterly oversimplification of the reality. And that's exactly what I tried to convey in my presentation. And actually, we know that. We know that. And I got inspired, and this is one of the images that I acquired together with Roger Tuttle more than 2 decades ago now, like 2.5 decades ago, where we physically flattened major parts of the cerebral cortex, of the neocortex of macaque monkeys. And in this case, you can see this here. This is a physically flattened part cut through extrastriate visual cortex. This is primary visual cortex. And you can basically cut from the pile surface to the great white matter boundary, different sections. So you get an overview throughout the cortical depth of many, many different areas. So here you see a lot of territory, extrastriate visual cortical territory. And it doesn't matter where exactly it is. But I want to draw your attention to these little, I hope it's visible on your projection screen. You see these patches, basically, these dark patches of activity. This is a deoxyglucose map. So it's basically a metabolic map, which has exceedingly good resolution and sensitivity. So you see dark patches, and within these dark patches, you see other smaller patches, big patch here, but with modular patches and so forth, which correspond to regions that are more active compared to the lighter gray regions. And actually, this pattern of activity with high metabolic activity and low metabolic activity and organized in very small patches we saw throughout the cortex, not only in extrastriate visual cortex like here, this is a lot of extrastriate visual cortex, but also in prefrontal cortex, parietal cortex, medial parts of cortex. We saw it all over the place. Unfortunately, at that time, we could not make any sense of it because the oxyglucose, you can sort of like label activity linked to one condition, 2 conditions. So within one given animal, you could only compare activity generated by 2 different types of conditions. So despite the fact that we saw this highly granular pattern of activated small parts of cortex, basically most often we could not make any sense of it. But it shows that cortex contains these small functional units, and they are not necessarily constrained to specific areas. Let me make my point further. So I call these units, and I shy away. Everybody would say, yeah, these are columnar-like structures, but I shy away from this connotation of columns. I think it has, whenever you start using the term columns, you get into controversies like what's the size, how many are there. People immediately think about columns, when you talk about columns, about orientation columns and so forth and so forth. They are very well organized, they have particular sizes, they have a particular frequency of occurrences in visual cortex that is very, very well studied. I think it is the things that we are seeing is much more different or variable compared to these traditional, for example, orientation columns in primary and secondary and higher order visual cortical areas. That's why I call this mesoscale functional units. It includes columns, it might include mini columns, it might include hypercolumns, it might include subaerial structures, not confined to the entire area, a particular cortical area that you're investigating. Just a few other examples. This is also from the time that I collaborated with Roger. Here you see the lateral view on a macaque brain, and we've literally flattened here, as you can see, major portions of extrastriate visual cortex all the way to the temporal pole. From occipital V1 is cut away here already, and then you get a flat map, and you see these dotted patterns here, which corresponded to regions that were activated by color versus achromatic gratings, in this case, of the same mean luminance. And you see this very patchy type of work, of activity related to higher activity for the colored versus the achromatic gratings, which stretched out into really anterior portions of anterior infratemporal cortex in this case. Of course, there are many more examples also from our work. Here, orientation columns, activity, deoxyribonucleose activity in the different layers of area V1, from the upper layers to the deeper layers, where you can also show that they're basically stretching throughout the cortex, which is much harder to characterize or identify using optical imaging and other techniques that are only sensitive for the superficial layers of the cortex. This technique literally showed that that these columnar structures stretch throughout the entire depth of the cortex. This is V1. This is V2. The same thing. Here we had two different labels in the deoxyglucose experiments, and when you overlay them, you see this very nicely interdigitated pattern of this, in this case, also of orientation columns. For here you see the red and the green orientations that we used. Of course, it was achromatic orientation, but you see these orientation columns. So you can go on. Many other people, of course, have shown these kind of structures. This is very nice work from Anna Rowe, also in area V1, part of V2, area V4. In this case, orientation columns again, orientation columns, but also here in V2, you see these small structures, much less frequent than the orientation columns, where you see these small subaerial, that is within V2, subaerial units that are responding to the axis of motion, yeah, which we also see to some extent for the orientation columns here in area V2 in panel F and G of this paper from Anna. We also know that infratemporal cortex has a similar kind of organization. In this case, also very influential work from Tanaka's group and colleagues that showed within infratemporal cortex an organization for features of objects that are clustered, where the neurons are clustered together, that are responding more or less similar to specific features. Yeah. These are functional patterns. This is sort of like what we know. But we also know actually for a few areas, for example, early visual cortex, that these functional modules are have very specific connections. And this is work, of course, that dates back decades. Marge Livingstone and David Hubel started with it, and many others have followed up on these smaller subaerial functional compartments. And they also showed that some of these compartments, for example, here, the blobs in the superficie, which are located cytochrome oxidase rich blobs in the superficial layers of area V1, basically are projecting very selectively to subcompartments in area V2. For example, the thin stripes, the oxidase based upon oxidase cytochrome oxidase staining in area V2, and then to specific compartments in area V4. The interblob regions, on the other hand, primarily project to the pale stripes in V2 and other subcompartments in area V2, for example, for layer 4C alpha, the part of the granular layers of area V1, then project to other subcortical areas within area V2, and from there, the thick stripes of area V2, and from there to, for example, empty V5. So you have these in early visual cortex. We know that we have subaerial, what I call mesoscale functional units, which are also anatomically connected. They form very specific networks, mesoscale functional networks. In other words, if you know function of area V1 and you don't make a distinction between these blobs and these interblobs, you basically don't know what is going on there, because you mix everything up, you mix up these connectivity parts, you only start to understand how this area works, or the subdivisions of this area work, by looking at the individual subcompartments of it. Yeah, so the thin stripes also project them to specific color selective regions within area V4, which have been called the color globs by Bevel Conway and colleagues. So mesoscale functional units in at least some parts of the cortex form mesoscale functional networks. Both these mesoscale functional units within an area are connected. Blobs are preferentially connected to nearby blobs, and also between areas. So they form very specific interareal connections. This is known for visual cortex, early visual areas in the non-human primate brain. Of course, we also know from many studies that exist in other parts of cortex. And I think a very nice example is in premotor cortex from Garbawi's group, who did optical imaging in this case, and who sort of like collected, this is a hemodynamic signal as well, collected optical signals from large parts of premotor cortex. And he did what a lot of people do in fMRI, for example, resting state optical imaging in this case. And when he seeded in a particular patch of premotor cortex, he saw correlated activity in fine patches, in this case a little bit more anterior, in dorsal premotor cortex. So you see this network. These dashed lines are areal boundaries. Again, it's not the entire area, but you get like functionally connected patches of correlated activity. The same thing in ventral premotor cortex. If you see it more ventrally, you see also patches which differ from these patches. The nice thing is that he also did more or less the same experiment, but instead using resting state functional connectivity, just correlated activity while the monkey is not doing nothing, basically, where basically the activity goes up and down during rest in a correlated manner. He electrically stimulated exactly the same spot as the one that he investigated here with resting state functional connectivity, and he sees more or less the same patches appearing. Of course, this is a stronger method because he stimulates and then he induces. So there is a causal relationship. This is correlational. Here there is a causal relationship between increasing activity in exactly this region and seeing evoked activity, electrically stimulated evoked activity in more or less the same spots that he saw here. Finally, I think he finished it very nicely by also injecting anatomical traces and the connectivity patterns, the functional connectivity. This is called effective connectivity patterns that he observed fitted surprisingly well with physically, anatomically connected patches. So when you inject the tracer here, you see that these patches that show up here are also anatomically connected, both for dorsal premotor cortex and for ventral premotor cortex. So again, the most important thing is you have this patchwork of subaerial units that form a connected network, in this case, across different areas. But these are still, as you can see, a relatively small number of areas, and this is restricted to a small part of, in this case, premotor cortex. What about prefrontal cortex? I talked about visual cortex. I talked about premotor cortex. What about prefrontal cortex? The standard view of prefrontal cortex is that it exists of relatively large areas, and some people even claim that there are no real areas, that is more like organized in gradients, where functions gradually change from the posterior to the anterior, or vice versa, of course, parts of prefrontal cortex. Of course, we also have the cytoarchitectonically defined areas, area 46, 8, 9, 10, and so forth and so forth. As I said, some authors claim that there's more gradients that span over different cortical areas, as you can see here. And in fact, there is very little information about subaerial functional divisions in prefrontal cortex. However, I think there is very strong anatomical evidence that these kind of mesoscale structures, of course, from anatomy, you cannot say anything about function, but that they might exist in prefrontal cortex. It dates back already many, many years here. Very nice work from Pat Goldman Rakesh, who made injections in very specific parts of prefrontal cortex, and the connected patterns were very, very focal. Yeah, this is just one example. I think another very nice example, and there are many out there from the Parma group, who also injected, in this case, in a very restricted part of area 46. And then they observed in remote parts of prefrontal cortex a very stripe-like, you could say, columnar-like pattern of connected areas. I think very beautiful work, which coincides with it, very recently published from the Japanese group, who injected, in this case in marmoset monkeys, both antro and retrograde traces. And you see here this very nice pattern, repetitive pattern of connected regions with the site of injection. And because they injected both antro and retrograde viral vectors, they could overlay these two patterns of connectivity, both the antero and the retrogradely labeled neurons and the anterograde labeled axon terminals. And you see that they coincide, very surprisingly, in a columnar-like fashion. So you have, when you inject in a particular region, you get these mesoscale anatomical structures. And on top of that, especially in the upper layers and somewhat in the deeper layers, you get like also more diffuse spread of mainly anterograde labeled axon terminal. But again, it showed that despite the fact that you do not have an idea what these things do and what the function of these mesoscale domains are, it indicates that also prefrontal cortex is probably organized in a similar way as, for example, premotor cortex and visual areas, as I discussed in the beginning of my presentation. Of course, anatomical evidence does not mean that they are functionally different, and surprisingly, for prefrontal cortex, there is very little, not to say no functional evidence. There is some evidence where people, for example, Constantinus, has shown that there is grouping of functional properties of cells that are located in the neighborhood, but they never showed maps. I think it's very difficult just with electrophysiology to prove that. Of course, David Jubel also showed that with electrophysiology, but to get a very clear overview of how these mesoscale functional domains are organized, you probably need imaging information. So the tenet of my presentation is that mesoscale functional units, basically, and again, this is an image in this case from the superior temporal sulcus, where you see these small dots in particular areas of the STS, that these mesoscale functional units form canonical, or actually the canonical building blocks of primate cortex. Of course, you have to show that it is indeed the case throughout the cortex. Very importantly, and not trivial at all, they should form at least functionally connected networks. Might be through multiple synapses, but at least these things should have very specific functional connectivity. And of course, imaging doesn't tell you anything about what the neurons are doing. You also want to see what the function is at neuronal level, whether at neuronal level these mesoscale functional units can be segregated. I think if you want to look at the entire brain, probably fMRI and high resolution fMRI is one of the prime techniques to do that. And of course, when you want to go to the neuronal level, you want to add electrophysiology to this so that you can target specific mesoscale functional units with your electrodes, which of course is also a non-trivial thing to do. So inspired by the deoxyglucose work that I did together with Roger 2.5 decades ago, we engaged in this endeavor basically, and more recently we started to implant MR coils on top of the skull of the animals, while also being able to have access to the brain itself. So we basically have integrated receive coils in the monkey, and then we do fMRI. We simply do fMRI, but with these implanted coils, and we have now animals with up to 16 channels that are embedded basically in the head post of these channels, together with integrated recording wells. And together also with contrast agents, basically, we get to a very high resolution imaging platform that we can use at relatively low field strengths. Actually, we scan at 3 Tesla. And this is sort of like an example. I think this is a 16-channel coil. So we built this on a replica, a CT. We take a CT of the skull of the animal, and then we 3D print it, and then we build these coils, monkey-specific coils, basically, for every individual. So they are permanently embedded on top of the skull of these animals. Another thing, and this is why I give this map, we also use contrast agents. We started to do that when we engaged in fMRI in awake behaving animals. We basically inject rusted iron, and that improves the sensitivity of our measurements by a factor 3, 4, 5, in addition to what we gain with these implant coils. And here, this is data from exactly the same monkey where we scanned, we did the BOLT experiment, exactly the same experiment as the day after, where we did the Mayan experiment using exactly the same stimuli. It's not even relevant what these stimuli are. And we acquired exactly the same number of images, and the monkey was working exceedingly well. And you obviously see the major change in data that you're getting with the Mayan and with the BOLT. So this is data that we acquired with implanted coils, and the resolution is 0.6 millimeter isotropic voxels in both the BOLT and the Mayan case. So what we do is we plug basically these animals. We plug these animals with implanted coils. I'm going back here, trying to, oops. We plug these animals in an MR scanner, and then we do a wake behaving fMRI. We basically do exactly the same as many people do, with the obvious difference that we have to motivate these animals, and that we have to reward these animals while they are engaged in a task. Whatever I'm showing today is a very simple task. They were simply required to fixate to a fixation dot in the center of the screen. Then we present all kinds of stimuli in the background. So the big difference is that we have to reward the animals. The other thing is that the monkey's head is fixed rigidly to a chair. And the other difference is that on top of requesting the monkey to fixate to the fixation spot, we also ask them to keep their hands, both hands, still in what we call a response box, and that reduces motion considerably. So the monkeys are not, they can move their body, but because their hands are in a response box, basically, and the reward is contingent upon fixation and keeping their hands still, body motion is very much reduced, and this has a very significant effect on the quality of the data that we are requiring. So, for example, just to relate what I'm going to show next with what I showed you in the beginning, we wanted to see whether we can visualize with this technique the color sensitive thin stripes in area V2. And we basically asked monkeys to fixate to fixation dot, and then we had fMRI block design where we alternated between colored in this case, red-blue gratings, which were also expanding and contracting, alternated with no stimulus condition, alternating with a condition where the same stimulus was shown, but in this case, achromatic, right? So we have a colored stimulus and an isoluminant colored stimulus and an achromatic stimulus, a very simple paradigm. We also did in the same experiment, basically, we used another stimulus set where we alternated between disparity defined gratings, exactly the same grating, but now not based upon color or luminance contrast, but based upon disparity, and we compared that with exactly the same stimuli, but without disparity defined grating. So it's sort of like the dots are all presented in the frontal parallel plane in this case. And indeed, this showed, and this is just, I'm going quickly over this, it is all published, or most of it is published basically, where we see in area V2, in the topically defined area V2 of one subject in this case, but we have it in multiple subjects, you see this interdigitated pattern of disparity selectivity and in green, and color selectivity in blue. And also we did other experiments where we looked at motion selectivity, is a little bit less relevant, but you see that green and yellow, this is literally T-score maps, this is thresholded here, the green and the blue, that the motion selective parts of V2 coincide greatly or mainly with the disparity selective patches. So indeed, we see this interdigitation of disparity and color selective patches, probably putatively corresponding to the thin stripes and the thick stripes in V2. So this motivated us, like, yeah, maybe even at 3 Tesla with these implanted coils and with contrast agents, we can get to reliable measurements of subaerial mesoscale functional units. This is just proof of concept. And then we went on. Are there these kind of mesoscale functional units? Can we identify? Can we look at them? Can we observe them non-invasively in the rest of the cortex? Obviously, a very important part of research has focused in extrastriate visual cortex, has focused upon category selective patches, for example, patches areas basically that respond more to faces than other object categories, to bodies more than faces, bodies more than other object categories, and so forth and so forth. Most often, these patches are considered relatively large and also relatively homogeneous. For example, we showed here many years ago maps. This is a flat map of a macaque cortex. We showed here several larger patches that are responding more to bodies than all kinds of control objects, sort of like the body selective patches. We all know, and you all know, the phase selective areas identified by Doris and Winrich and many others, basically. These larger patches from the posterior part of exostriate cortex to the anterior pole, in this case, of infratemporal cortex, relatively large and homogeneous phase selective areas in this case. So we said, okay, let's do this very simple experiment and do a category selective localizer where we presented to the monkeys a bunch of stimuli, different categories, bodies, headless bodies, monkey, human, objects that are matched, for example, in aspect ratio for monkey bodies or for human bodies. These are more long stretched than monkey bodies, monkey faces, human faces, animals, which do have faces and which do have bodies. Birds, same thing. They also have a head with eyes and a beak and a body, and then fluids and sculpture. Sort of like we presented this to these animals and embedded it in pink noise. A very simple experiment, a block design. Well, we did both block and event-related design, but all the data I'm showing was from a block design fMRI experiment in monkeys with this implanted coil and also with contrast agents. And just to give you an idea what we are getting, this is just throughout, this is occipital cortex. You see the coordinates here relative to the interaural plane, from more posterior to more anterior. Body sensitive areas near the STS and also parietal cortex. And I'm going back. If you see our data, this is a flat map, but more or less this is the STS, here the IPS. We basically did not see, this is low resolution imaging. I think it was like 125 millimeter isotropic voxels in this case. Here we are talking about 0.6 millimeter isotropic voxels, and you see a much richer pattern of activity for bodies compared to monkey objects, the control condition, including parietal cortex, and I will also show you later on some data for frontal cortex. Very small patches, sometimes bigger, but also some smaller. Here you see 1, 2, 3 patches. And you see the threshold here is quite liberal. It's not very conservative. If I would see a pattern of activity like this, I would not believe that these small things are very relevant, given the statistical threshold. I only believe that it becomes real when you do it the next day and you see more or less the same activity patterns. And I want to draw your attention to some of these patches you see here. These are coming back. These things here in parietal cortex, they're coming back. Here, for example, this is in the STS. We saw very consistently body selective fMRI activations in the lower part, layer 6, layer 5 maybe, of this part of the STS. Same thing here, more towards the upper bank of the STS. Very reliable, very small patterns of activity that we could see. So this was very engaging. I also must say, you see that the activity is constrained to the cortical ribbon. We didn't do any masking of cortex and things like that. This is literally what you get. Very simple, straightforward GLM-based fMRI activity, very well confined. The reason why we see these small things is probably because activity is concentrated to very small parts of the cortex, and we don't suffer from partial volume effects. If your voxels are big, you're of course going to smooth activations, small activations, by throwing in noise from neighboring regions that have nothing to do, in this case, with processing body information. Another example here, in parietal cortex, you see these 3 different spots that are very reliably identified in the same animal, of course, in 2 different days. Just to give you a zoomed in version, here you see these deeper activations in this part of visual cortex. So again, same thing, body selective activity one day, body selective activity the other day, and you see even the small activations, body selective activation, sort of like what I call, it is definitely at superaerial level. Here the blue outline is an area, it's part of LIP, and you see that within LIP you definitely have like multiple spots that are segregatable and reproducibly identifiable over different days. Sometimes you have bigger activities here, yeah, and I will come back to that later on. So we see highly reproducible fine-grained patterns with these high-resolution fMRI techniques. You could also argue, yeah, maybe because like in bold, you have the effect confounds, potential confounds of large draining veins. Maybe these are the regions where you have large draining veins. First of all, with our contrast agents, we don't have that problem. We basically black out the big draining veins. We simply don't suffer from it compared to regular bold imaging. But still, you could say these are maybe the regions that you see here of cortex where we have like higher sensitivity. And this is body day one. And you see, this is the same animal, different experiment, body disparity. And you see that the activities really jump depending on the experiment. So it's not that it's restricted to regions where we have like higher sensitivity. On the other hand, you see here, for example, in the IPS, in the interparietal sulcus, a spot that is active for bodies versus objects, but that is also active for disparity versus a flat screen, basically. Most of the cortex is different, but there are a few spots where information probably is being integrated, and it would be very interesting to see whether we would have here more selectivity for bodies if we would have like 3D instead of two-dimensional bodies. And again, highly reproducible disparity maps across different days in the same animals, as you can see here in this map. This is faces and bodies, more or less the same story. In this case, body selectivity is coded in blue, face selectivity in red. You see this pattern of activity, quite a bit of body selectivity in interparietal sulcus. A little bit we see sometimes also in the parietal sulcus face selectivity. Again, highly reproducible. I will zoom in a later section here. You see face, body, face, body, face, body. Highly reproducible across different days. Here you see it also, face, body, face, body, face, body. Sort of like a pattern, if you run across the cortical ribbon, basically, that is mimicking what we saw in area V2. You alternate between selectivity for one stimulus category versus the other, faces, bodies, faces, bodies, faces, body, that you can see in an alternated version here. And here you see it, the zoom in version, faces, bodies, faces, bodies, faces, bodies, which again is highly reproducible between the two different days of scanning. Yet we also see, we still see regions here, for example, which is big, which are large, which are in this case selective for faces, and which seem to be, of course, here you're only using a very limited set of stimuli, faces and bodies, to identify this spot. But are these areas really heterogeneous? So for the large patches, for the large category selective patches, we address the question whether there might be within patch heterogeneity. And we used to assess that issue, we used the entire functional profile, sort of like the fMRI tuning in this case of these very, very small voxels that we are acquiring. We looked at the tuning of the clustering of these tuning profiles of these very small voxels, and also whether potentially, if there is subaerial clustering, whether these subaerial clusters also have different functional connectivity. So basically what we did is a very similar experiment as what Doris did. So we identified areas, for example, phase selective area ML here, relatively in posterior ventral stream of the monkey, based upon relatively strict conditions where activity for faces has to be larger than for fruits, for objects and bodies, and across different days. So relatively conservative. Really, we wanted to have voxels that are really selective for monkey faces. And then you could think, yeah, if they are selective for monkey faces, it's a very homogeneous area. Of course, this is done, in this case, Doris used voxels that were 2 cubic millimeter in size. So we did the same thing. We used the same experiment that I explained before, but now with voxels that are about 1 log unit smaller, voxels of 0.22 cubic millimeter. And we presented the 10 categories, as you can see here, that I presented earlier. Monkey faces, human faces, monkey bodies, human bodies, animals, birds, sculptures, fluids, and then other objects. And obviously, these voxels in ML are phase selective. They respond more to monkey faces and human faces than any other category. A little bit to birds and animals, but of course they also have to some extent faces, but they're definitely phase selective. So the next thing that we do, and it looks relatively homogeneous, but then we did like unsupervised hierarchical clustering, and we found consistently 3 different clusters. So these voxels, these response profiles, basically can be clustered in 3 different clusters, in this case for area ML, a pink one, a green one, and a blue one. And you see that indeed, when you do this clustering, there is some variability. So the first pink cluster is phase selective, is probably the strongest phase selective cluster, responds only mildly to these animals and to these birds, and basically not much to the other categories. The green cluster, voxels, basically, these are all voxels. The rows are all individual voxels, 0.22 millimeter, cubic millimeter voxel. They start to respond a little bit more to these animals and birds, but they are still phase selective, of course. The blue voxels here, in this case, labeled blue, they are also phase selective, and they started to respond more and more to these animals and these birds. When you average them, you get like 3 clusters, the pink, the green, and the red. And basically, this is exactly the same information, but averaged across all the voxels belonging to the pink, all the voxels belonging to the green, and all the voxels belonging to the blue category. Of course, then it becomes interesting, most interesting, where are these voxels located within that area? Is it a salt and pepper kind of organization, or is there a spatial structure? Because of course, functional clustering does not mean that there is anatomical clustering. So we back projected it to the brain, both in the left hemisphere. Okay, these voxels were from both the left and the right hemisphere. And to our great surprise, we see clustering here. We see spatial clustering, pink, green, and blue in coronal slices, both on the left and the right-hand side. And when you look at the sagittal thing, we also see this kind of blue, green, pink, green. So there is a spatial clustering that we could retrieve in area ML. So large, seemingly homogeneous areas can be subdivided in mesoscale functional units, what we call mesoscale functional units, which have different functional profiles. Yeah. Of course, the next question is, like, do they also have different connectivity profiles? And actually, we try to address this question by looking simply at interhemispheric. This is a large distance, of course, interhemispheric functional connectivity between these different clusters, the pink, the green, and the red. And the basic question is like, are the pink voxels in the left hemisphere preferentially connected, functionally connected with the pink ones in the right hemisphere, more so than with the green ones in the right hemisphere and the blue ones in the right hemisphere? Yeah. To do that, we used functional resting state functional connectivity analysis. We used the same animals, and they were scanned at the same resolution. They didn't have to do anything. There were no stimuli shown. So it was a completely independent experiment, basically. And we looked at resting state functional connectivity. And actually, to my great surprise, I was never a big fan of resting state functional connectivity. But to my big surprise, there was a difference between within cluster 1 to cluster 1, cluster 2 to cluster 2, cluster 3 to cluster 3, within cluster functional connectivity compared to across cluster connectivity, cluster 1 to 2 and 3, cluster 2 to 1 and 3, etc., etc. Of course, you can do a little bit more sophisticated things on this connectivity business. These clusters were not equal in size, so you have to control for it. And when you do sort of like a permutation testing, you basically see that the random distribution of functional connectivity definitely differs very significantly between the within and the between clusters, basically. So it's a very robust finding. It's not a big difference, but it is different that the functional connectivity between the different clusters in the left and the right hemisphere is different. We also did the same trick, and I can go quickly here, for another category selective area, a body patch, which is more selective for body stimuli compared to other stimuli, and that's what you see here. It's a smaller patch, so we have like smaller voxels, but we also got 3 different clusters here. And obviously, they respond more to bodies, monkey bodies and human bodies than anything else, but they also respond to some extent, again, to animals and birds. But of course, they also have bodies. There are slight differences, sort of like the
Yeah, I think Wim got disconnected. कोई मैसेज भेजे कि shoot. این مشکل از اونه چجوری اینا هی دیسکانکت میشن
I believe this is a problem on the part of our speaker, and we will resume the talk as soon as he connects to the call.
Okay.
Oh, okay.
Hello. Did I lose you?
Hi, Wim. We lost the connection. Now we have you back.
Ah, yeah. Okay. I'm sorry. And wait a minute. I have to share my screen. Where did you lose me?
Um, I think you were talking about hierarchical clustering within the body patch.
Ah, yeah. Okay. And already the
And what?
cell stuff. Well, I'm going back to my presentation, and then you have to guide me where I lost you. Wait a second. Did I lose you here? This, hello. Do you see me?
Yeah, I think no. Go back. Yeah. It was there.
Okay. All right. Sorry. Yeah. Probably network issue or whatever. I think it was clear that we have functionally defined subclusters within a larger area. In this case, area ML, right? And you hear me and you see me? Because I don't have any feedback basically. Reza, do
Say
you
it
hear
again.
me? Do you hear me?
Yeah.
Okay. And you see my screen, right?
Yes.
Okay. So the thing is, like we have these. I'm going back. We have these functionally defined subunits within area ML, phase selective area ML, and we simply ask the question whether units, MFUs, mesoscale functional units of the same type, are preferentially connected across different hemispheres, the ML in the left hemisphere and the right hemisphere. In other words, whether the pink voxels are strong or functionally connected with the pink one in the other hemisphere, the green ones with the green ones, and the blue ones with the blue ones, rather than the pink ones with the green ones with the blue ones. These voxels are in very close neighborhood, so it's a very tricky question. I would never have believed that this would work. And to that end, we performed a resting state functional connectivity analysis, and in the same monkeys, obviously, also at high resolution with like 0.22 millimeter voxels, but the monkeys were just fixating, were not shown any of these stimuli, just resting state functional connectivity. And basically what we see is that indeed clusters of the same type in the right and the left hemisphere are more functionally or stronger functionally connected than clusters of a different type. This is just straight comparison of functional connectivity of the within cluster functional connectivity and the between cluster connectivity, and it is significantly different. And then we did also permutation testing, and basically we show the same kind of results. This is for phase selective area ML, but we also get it for a body area. This is body area where we use the same stimuli, and you see obviously now they are not phase selective, but they're responding more to monkey bodies, human bodies, animals, birds. We also got tree clusters, and when you map it back to the cortex, we also get like spatially segregation of these voxels that have a different functional profile. We also showed for MFB, exactly as for the phase patch, that the within cluster connectivity, the pink in the left and the pink in the right hemisphere, they have like stronger functional connectivity than, for example, the pink with the green and the pink with the blue in the two hemispheres. Yeah. So basically, we see in another area, in this case, a body selective area, the same kind of a difference in functional connectivity. So we not only can segregate a larger area that is seemingly homogeneous functionally in subparts, we can also identify that these subparts have at least slightly different functional connectivity profiles across long distances. This is from the left to the right hemisphere. The nice thing about this body patch is that in other animals, basically, we recorded single units using exactly the same stimuli. Different animals, same area, but now we had single unit responses from this region. And that's exactly what I'm showing here. This is the matrix. Now the rows are not different voxels, but different single units. And we did the clustering here, the same clustering with the same criteria. And we also got 3 different clusters. Again, a red or a pink, a green, and a blue cluster. Of course, the interesting thing is that you can then try to relate these single unit clusters with fMRI clusters. This is the average of the MRI cluster 1, average of fMRI cluster 2 and 3, and below it, single unit cluster 1, single unit cluster 2, and 3. So you have completely independent data, even in different monkeys, and it is obvious that these clusters are looking reasonably similar, and when you correlate them, they are highly correlated. So whatever we see with high-resolution fMRI, hemodynamic signals, basically, we retrieve also in single units, even in a different animals, which basically probably means that there is this kind of cluster really at single unit level, in this case, area MSB. Obviously, it's not easy to have also the single unit evidence from other areas, because this is a very painstakingly experiment. At least we know with high resolution at population level, we get the same thing with single unit recordings. I'm still online, Reza? You still hear me?
Yeah, we can hear you.
Okay. So I continue. What about prefrontal cortex? I showed you in the beginning anatomical evidence for smaller subaerial domains in prefrontal cortex. Do we see that functionally? And I can go very quickly. I don't think the movie is working, so I'm skipping this. We did also other experiments, for example, optic flow, and where you have random dots that are sort of like rotating clockwise, counterclockwise, that are expanding, contracting, very low-level optic flow stimuli. And to my big surprise, this is again coronal sections through, in this case, frontal cortex. You see here in dorsal frontal cortex, probably SAF, you see these patches that are differently activated for expanding compared to rotating patterns, both on the right-hand side and the left-hand side. Not a lot. It's not what you expect based upon what you know from orientation columns in V1, where you would expect many. Well, first of all, it's a big thing that you see these things, but it's not that you have like 100 of these mesoscale functional units there. It's just a few within an area. This is near FEF, more or less the same thing. This is one day, odd days, this is even days, highly reproducible pattern. I think I can zoom in. You see in 2 different days, highly reproducible patterns in frontal cortex. This is an occipital cortex. We more or less see the same kind of thing. Of course, the big question is like, again, are these blue guys there, the expansion, rotation, contraction kind of mesoscale functional units, in this case, in parietal, in occipital cortex, are they functionally connected with the blue guys in frontal cortex? But again, evidence for definitely subaerial, highly specific functional profiles that we detected with high resolution. This we confirmed very recently, and I'm just going over it very quickly, with focal ultrasound imaging now in parietal cortex, with it's non-invasively also CBV, but this gives us actually even much higher resolution. We can scan now from a restricted part of the cortex at voxel resolution of 0.011 cubic millimeters, so even 10 times smaller than what I showed you before. And this is very preliminary data that we just collected a few days ago. For the same stimuli, expansion, contraction, we see indeed in parietal cortex confirms what we saw with fMRI, very focal activity. This is single trial CBV measurement, and you see highly reliable signal, basically, and a difference between, in this case, rotation and expansion contraction for one voxel, 1.01 millimeter voxel. But again, it's not a lot. It is not a big area. It's a very, very restricted part, in this case, of dorsal stream cortex in the back of the brain. But again, it's an independent method to confirm that these small units do exist. Back again to prefrontal cortex. Again, very small, well, very low level experiment. Disparity. We see fine-grained disparity selective activity in frontal cortex, which I would never have betted upon until we did the experiment. This is what you expect. This is what you expect in occipital cortex. This is not what you expect in frontal cortex. Complex stimulus categories, faces and bodies. Again, you see this highly refined granularity also in prefrontal cortex, in this case for faces and objects. Sort of like more or less what we saw in the back of the cortex. If you have high resolution, you see this fine granularity, highly reproducible face selective domains in prefrontal cortex. This is the anterior pole of prefrontal cortex. Scenes. We did the same experiment where, and this is published so I can go quickly over it, different types of stimuli, including scenes, different types of control stimuli for the scenes, 2 different experiments. Also in prefrontal cortex, again, you see these very small subaerial scene selective areas here, in this case for 2 different animals. Again, for me, this was completely unexpected, given the fact that there is very little evidence from human imaging, for example, that this would exist in prefrontal cortex. Again, there you could ask, this is functional data. There's a correlation between a hemodynamic signal and, in this case, scenes versus non-scene stimuli, which we reproduced across different experiments. Are they also preferentially connected? Are these small things preferentially connected relative to other category selective regions in prefrontal cortex? So we have patches here in blue. This is a flat map of the entire cortex. In red, scene selective areas. In blue, in this case, just body selective areas in prefrontal, parietal, and temporal cortex. And also there, the functional connectivity based on resting state functional fMRI within a category selective network. In this case, scene selective network is higher, different compared to connectivity between the scenes and the bodies. You see a very big difference, even despite the fact that some of these body areas are much closer to the scene frontal areas than scene selective areas, and in the front of the cortex and the back of the cortex.
Sorry, Wim, for interruption. I think, yeah, if you can wrap it up in a few minutes, it would be great.
Yeah. The last thing is that we recorded in the frontal patches, and this is sort of like the last interesting bit of information. These are single unit recordings in these small face areas in the front of the cortex, right? This is single unit responses, different for faces, more for faces than any of the other categories that I showed you before. And this is an infratemporal one recorded in the same animal. So of course, with functional imaging, you don't have the temporal information that you can get with single units. And we see a very surprising result, basically, that here the red curve is the response for the same stimuli in the same animal, basically, for a frontal area, face selective area, and the blue one is for anterior medial infratemporal area. So the anterior medial infratemporal area is significantly slower than one of the frontal areas. So it cannot be that this area is sort of like steering or driving the frontal area. So there must be parallel stream going directly to frontal cortex and then maybe feeding back to anterior infratemporal cortex. But the fact that we see this doesn't mean that face selective signals propagate from back of the cortex to anterior IT and then to frontal cortex. Either there are parallel streams or independent streams for phase selective cortex. So I think that we have to go away from this scheme of areas, connected areas. We know basically that in early visual cortex, subunits are connected and they convey the information. They really form selective pathways for motion, for disparity, for color, for example. We know that. I think this can be extended throughout the cortex, and that you get like very selective streams of information flow across the entire cortex, including mid-level region and frontal regions where mesoscale functional units are the prime carriers of information, and that the areas actually are very, very irrelevant. So to wrap up, Reza, I think I showed highly reproducible mesoscale functional units in different parts of the cortex, very fine-grained for body, face, scene selectivity. Also, the larger patches can be subdivided. They are not homogeneous, and they show also consistent cross-hemispheric functional connectivity. And a functional connectivity between mesoscale functional units of the same type is different from these of different types. So I think the functional organization of category selective cortex is much more complex than proposed by most current models. And I propose a novel view on primate cortical organization in primates, which is based on parallel brain-wide mesoscale functional networks instead of interconnected areas. Thank you.
Thanks a lot, Wim. Now we can have a Q&A. We have time for some questions. It's actually quite surprising that you can see very fine structures near each other, given the spatial blurring of fMRI.
Yeah.
And also when you have very small voxel sizes, the signal is very noisy, yes, and still you get reliable distinctions in very adjacent regions of cortex, yes?
Yep, I fully agree. Of course, we have a few tricks to increase the sensitivity. I think it all boils down to resolution. I think it is the smaller the resolution, or the better your resolution is confined to the real structural organization, right? The less you will be bothered by partial volume effects. So you will gain actually by going to higher resolution. And then, of course, you also need to have the sensitivity. And I think we have the sensitivity by the combination of these implanted coils and the contrast stations. Obviously, things that you cannot do easily in humans. In humans, you probably have to go to a high field, maybe very high field, to get the same kind of data. The thing is that I'm quite confident that this data is true because we have independent information from other techniques from the single unit, of course, just from a restricted part of the cortex by definition. And now we start to do this ultrasound imaging, also from restricted parts of the cortex, but also conveying more or less the same imaging. So I think it's a combination of multiple things that you can look at this, Reza. I think behavior is also very important. So our animals are super well trained. I can show you data, raw data from APIs taken on the scanner, and they are very, very stable. And I think this also contributes to the quality of the data.
Okay, thanks, Pim. Is there any question? If not, I can continue asking questions.
Yep.
Uh, so, Wim, I have kind of a general question here. So, in early visual cortex, for example, in V1, we have these patchy organization like blobs and interblobs because they are they are supposed to cover the entire visual field. So basically, for each location in V1, we need color processing, we need disparity processing. So basically, these structures are repeated because they are going to cover the entire visual field. Okay,
Yep.
but I don't know what is the advantage of having a patchy organization in frontal cortex. Like
Oh, yeah.
you're going to have face body face body face body patches. What is the computational advantage of having such organization?
Yeah. I want to. This, of course, is a very tricky question. This is sort of like an empirical question that has to be addressed. First of all, I think the organization that we see in parietal, prefrontal, frontal cortex is not identical to what we see in V1. As I said several times, the size and also the number of these structures is vastly different between frontal cortex, for example, and V1. We have indeed exactly because of the reason that you're explaining, and we have like many more orientation columns because we have to combine space with a feature, right? Every feature has to be represented in every part of the visual field. We don't need that in frontal cortex. On the other hand, you could conceive that specific parts of information are transferred from even low-level information is still transferred to frontal cortex, where there is not yet, at least initially in the responses. For example, that last data set that I showed you, where one of these frontal PA phase patches respond much faster than AM, for example, in infratemporal cortex, that there is still a relatively low level representation of faces. You know, it might be a face, it's not a face, it's a color face. So before everything else that needs to be processed about identity, emotional content, etc., that you still have like relatively low level information in prefrontal cortex. Because sometimes the same stimulus, and I think it can have multiple reasons, sometimes the same stimulus needs to be used for very different outcomes, right? Remapping. So you have a very simple stimulus red that has to be used to stop or to do something else, or to, like exactly the same retinal stimulus has to be used in very different manners. And this is decided in prefrontal cortex. It's prefrontal, well, most likely prefrontal cortex that sort of like decides, okay, this retinal stimulus has now to be used in this and this and this context. So you want to have at least some level of segregated representations for relatively low level stimulus features in prefrontal cortex. You don't probably need the space. You don't need to know, okay, this red dot is presented at exactly this location in the visual field. So I think it's definitely different in terms of computation in prefrontal cortex and the back of the cortex. Obviously, the receptive fields are larger, although we also see for the faces relatively small receptive fields in prefrontal cortex. Yeah, I hope that answers your question, Reza.
I mean, to some extent it answers, but still, it doesn't explain why we have multiple patches. Like we could have, let's say, we could have one patch in PFC, one face patch, and that patch handles all kinds of processing for identity expression. I mean, why we need this replication of one patch? Like in IT cortex, you showed like bunch of face and body patches, like face, body, face, body, face, body. Why we have such organization? I mean, it could have been like just one face and one body, and that's it.
Yeah, yeah,
Why we need
indeed.
organization?
Yeah, it's a very very valid question, but I'm going to try to answer that question by that one data that set that I showed for parietal cortex, where I showed body selective small multiple patches and disparity, completely different stimuli, and most part of that stretch of interparietal sulcus, basically, and IPL. They are segregated, but in some of them they converge. Obviously, our stimulus set is limited, and it may be that it's sort of like to dynamically fuse different data sets so that you get like multiplexing in some of these patches that okay, this patch is really interested in not just processing a part of a body or a full body, but it is also interested in the three-dimensional structure of a body, while the other neighboring body patches in that part of parietal cortex is less the case. We know that this actually even happens at single cell level. So it might be that that there might be a computational advantage to have this also at at a level where neurons are clustered, right? I don't believe it's just one neuron that will do the job. It's probably several neurons in the neighborhood that are doing this kind of job in parts of cortex. Obviously, I think this is a very valid and very interesting question. The fact that we can start to study it, Reza, I think, because I would not have believed this until I saw it. That we can start to study it in great detail is already highly interesting, I think. Yeah. I see in the chat. Wait a minute. But I have
Okay.
to. Wim, you showed in your last slide that some information emerged in PFC sooner than 90. It was not body. This was face information. This was face selectivity. Yeah. Yeah, sure. Um, um, I'm actually writing a grant right at this moment to do this, but the problem is with focal ultrasound imaging. It's very nice and very. I think it's like phenomenal. I think it's gonna. I might be doing focal ultrasound, instead of fMRI in the very near future. The thing is, you also need to be. It needs to be accessible, and ultrasound only has like a particular depth penetration, so it's easier to do this from dorsal regions. Of course, interfrontal cortex, definitely the stuff that is near the ear, is very difficult to reach by these kind of technologies, both by optical imaging. Remember the difficulties that Tanaka and people had to get to really ventral parts of the infratemporal cortex. So I think in first instance, I want to go to parietal cortex and frontal cortex, where I think you can do that simultaneously. I think the beauty of focal ultrasound is also that you can much easier integrate it with electrophysiology and other types of technologies. You can online, you don't have to worry about all the MR compatibility and so on and so on. So I think it's a fantastic idea, technically not that trivial. And I think that some part, to come back to the other part of the question, that some part of prefrontal cortex is also to, you get information, what do we need to do with this information very quickly? Sort of like, I think, the Moshe Bar sort of like way of thinking, that you have like a very rough kind of idea of what it is that is presented on the retina, and it might be a phase, it might be not a phase, and things like that, and that sort of like low resolution kind of information is then fed back to more occipital cortex for more detailed integration. It might be predictive coding. I'm not sure. So I think we definitely should go away from the idea that prefrontal cortex is just sort of like the super high level semantic description that we know exactly what a face is, what mood a face is, what gender it is, and what kind of information a face conveys. It might be both.
So for those who didn't follow the question, so the question was about doing focus ultrasound imaging simultaneously from IT and PFC to answer some questions. So actually, I want to do a follow up on this question. Can you elaborate on this technique? Is it invasive, or I mean, you need to remove skull, or no? You can do it.
Yes, yes,
Ah,
yeah, yeah. That's the thing. So the data that I showed you was with the linear. It's you don't have to stick it into the brain. It's on top of the brain, but the skull attenuates basically the ultrasound waves basically. So you have to make a craniotomy. Uh,
you need
there
a
are
craniotomy.
yeah, you need to have your basically what we have a recording well, and then you basically put the transducer right on top of the cortex, so it's also limited. You don't have the full overview of the brain, but it is yeah, the sensitivity and the resolution is unbelievable. It's simply unbelievable. You know, if you have like access both of the transducer and an electrode, you can basically do things together. You can have like it's at the end of the day you're measuring also a blood volume change, so it's just basically like Mayan fMRI, but you can also have you can visualize your electrode. You know exactly where it is relative to the cortical layers and so on as well, and you can do electrophysiology simultaneously. So I think it has fantastic potential. The caveat is it is local. It's still local. People are working on two-dimensional arrays so that you can like image a volume rather than a slice of the brain. But yeah, the caveat is that you need to have a craniotomy. The thing is that people are also working on you still need a craniotomy, but to have materials that can replace the skull and that are basically not attenuating the ultrasound signal, so that you can basically remove the skull and then quickly put something else in place, that you don't have an open craniotomy for a very long time, which of course is also tricky because you get infections and so on if you don't take care of it.
So it measures blood flow, not neural activity. Yeah.
No, it's measuring. It's actually a Doppler effect. It is actually literally the movement of the brain. There are a few studies in rodents that showed a very high. It's much more spatially specific. So you're really looking at parenchymatic tissue, and in rodents at least, the correlation between what you measure simultaneously with the CBV, cerebral blood volume measurement, and neurons is surprisingly good. But still, it remains a hemodynamic signal, so it still is not fast and so on and so on. But I think to guide things, injections, electrodes, and so on and so on, I think it's phenomenal. To really know, okay, now I'm recording from layer 6 and so on and so on. We know this is a very, very tricky thing to do, and I think this technique can do that.
Okay. So you wanna ask your general question?
Uh, Thank you for your insightful presentation. We have two general questions actually that we want to ask all of our speakers. The first one is whether you have any piece of advice for young researchers in the field of neuroscience. And the second one is that how women have contributed to this field of study.
Well, I'm gonna start with the second one. As I data that I showed you, like I think 3/4 of the data have been acquired by a female brilliant study. Unfortunately, she left science, so she submitted one. So she did her PhD in my lab, and then she submitted a grant to get postdoctoral fellowship. She failed, which I think it was incredible. I think I still don't understand why reviewers did not the reviewers of that grant did not accept her, but she was so yeah, you know, disillusioned that she basically went to company to do data science. Of course, more money. It's somewhat easier life. Also, a lot of the people actually right now in my lab, I think have more women than men. It remains a problem for many reasons. I have seen it several times in my own lab. It remains a problem to get females finally into the PI at PI level. Why that is, I don't know. I have had several brilliant females in my lab who at some point quit for, I think, mainly professional, well, private reasons. But I think it's a shame. It's definitely a big shame. And the other question was, oh, I forgot. It was the female.
No, yeah, the other one was general advice.
Advice, general advice.
Yeah,
Um,
for young researchers.
yeah. uh, don't don't necessarily take the easiest route, right? I think perseverance, even and science is not easy, right? And I think you probably, and I think this is a big problem. Junior people in science think that everything has to be successful. This is not the case. This is not the case, and it is by perseverance. It's sort of like by keep, by being a trooper, by keep trying, that you will succeed. Everybody who says that, yeah, whatever I did was successful is a liar. And this is the thing that I see in my lab, where junior people do some great stuff, and then sometimes, yeah, the things don't work out as you think, for many reasons. This can be technical reasons. This can be simply because our hypothesis was wrong, and so forth and so forth. That they are getting demotivated and things like that. And I think this is big part of science. It is not by definition, because nobody has done whatever you're doing before you don't know. You need to be open-minded, right? Even if you think that whatever you're doing should work out, it may not be the case. This is exactly the fascinating thing of science, is you don't know the outcome, so you still have to do the experiment. And sometimes, you have to say, yeah, I was wrong. It's my hypothesis was wrong, but still the data that I'm getting is very, very useful, and it changes our thinking of how the brain works. And the other thing is, like, don't be too dogmatic. So sort of like think out of the box. Sort of like if you see data, it's sort of like, how can I explain this in another way than other people are thinking about this. I think science in general suffers from dogmatic thinking. People think that they know how things work, which may not be true, right? So you sometimes have to look at it differently. But that said, I think science is the greatest thing you can do. It is, by definition, something new. You can be high level surgeon and doing fantastic thing and helping patients, at some level it becomes routine, right? You will do the same surgery, everything is different, but it will become routine. By definition, in our job, that's not the case. It is not the case. You're doing things nobody else has been doing, and I think it's just the most fantastic job there is on earth. And it's always stimulating. You have to think. You have to think about. It is. I enjoy so much reading beautiful papers from other people. We usually lack the time to read everything, but it is so inspiring to see it. Motivation and perseverance, I think, is a big thing.
Thanks a lot, Wim. So we can finish